everyone, you're listening to the TechFest podcast, and I'm Raga, and we're back with a new diabetes episode with John today for all the science lovers and curious minds. Hello, John.
Hi, everybody.
Hi, so how are you?
I'm very good. Thank you very much. How are you?
I'm great. John, would you like to give a bit more intro about yourself to our listeners?
Sure. Yeah. So I'm John Barrow, and I'm a senior lecturer at the University of Aberdeen, where I teach quite a lot these days, actually, rather than doing too much lab research. Um, but my teaching is largely focused on biochemistry and molecular biology. So really understanding how all of the small molecules and bits and pieces in our cells function, and my research background is actually in diabetes and understanding how the various genes that control pancreas development are actually regulated, and then maybe what implications that might have in terms of treatments for the condition.
Great. So let's get started with the questions. What processes down the pancreas development?
In terms of the pancreas and how it develops, it starts off in the early embryo as just a simple gut tube. So as the embryo develops, there is literally a very simple tube that runs from the top to the bottom of the embryo. And in the early stages of pancreas development, there's various signals and um, things like growth factors that regulate what that gut tube is going to become. So obviously there's other things that other than the pancreas that form parts of your digestive system. So all of those things also come from this gut tube, or most of them do. In terms of the pancreas, though, there's a set of signals that tell a region of cells within the gut to become the pancreas. So often in the early stages, that's growth factors, things like retinoic acid and other kind of chemical signals. And then later on, when that process starts to happen, what you get is if you can imagine this tube expanding out in two directions, almost like a kind of tree, and branching ever more and more branches that form all of the other cells of the pancreas. So in your pancreas, you have largely digestive enzymes that are produced by what's called the exocrine pancreas. So that's the majority of the pancreas, actually. About 95-98% of it or so is that.
Talking about pancreatic enzymes, isn't pepsin a part of it? As far as I remember from my biology lessons.
So that's right. Yeah, the pepsin is part of digestion, but pepsin's actually an enzyme that's found in your stomach. It's not made in the pancreas, so it works just in the stomach. Pancreatic enzymes are something else.
Mm-hmm.
The bit I'm interested in is called the endocrine pancreas, and that forms, just a few percent actually of the whole organ, and and that's the islets of Langerhans. So they're little clusters of cells that contain different cell types, but the one that's really important for diabetes is actually the beta cell, as it's called, and that's the one that makes insulin, which is really important in diabetes and regulating blood sugar. So that process of getting from that gut tube to all of these different cells, be them the exocrine cells that make digestive enzymes, the ducts that carry those digestive enzymes, or the islets of Langerhans cells, all of that is controlled by a series of transcription factors, which are proteins that regulate particular genes.
Mm-hmm.
And those genes are then what dictate what that cell is going to become. So it's almost like a series of checkpoints that you go through from an embryonic gut to cell at the first stages, and then all the way through to lots and lots of different cell types later on in development.
Mm-hmm. So pancreas has different enzymes, and part of these enzymes is insulin, which plays a big part in diabetes. So that's how we can get started with, is it?
Yeah, that's right. So I think the enzymes digest your food, but then the endocrine part of your pancreas, these islets of Langerhans make, is not enzymes, but it's actually hormones that regulate various processes in our bodies, insulin being one of them.
So, can you tell our listeners a bit more about the area you've been involved in research about diabetes?
Sure, yeah. So really, I mean, there's a few different aspects to the research that I've done in the area of diabetes, but probably the main one was actually trying to understand some of the processes that regulate how the islets of Langerhans develop, um, and focused actually on one particular gene, which has a bit of a strange name for a gene that's found in the pancreas, but it's called neurogenin 3.
Ah, sounds like a brain one.
It does, because it's also produced in the nervous tissue as well. So actually, weirdly, it's expressed in the brain and also in the pancreas as well. But it's a really key gene for regulating the endocrine part, so the bit that becomes these islets of Langerhans, these little clusters of cells.
Yeah.
And the reason for looking at this and understanding how this gene may regulate other parts of that developmental process is to really ultimately try and find a cell replacement therapy for diabetic patients. So, if you're going to go and put cells into somebody that are going to be replacing these beta cells that in a diabetic patient don't maybe work very well or are non existent, then you want to make sure those cells are as close to a beta cell as you possibly can make them. And one way is to understand the natural process of development, and then try and mimic that maybe in things like stem cells, for example.
Okay, so you take beta cells from an embryo. Is that stem cells research or something?
Yeah, so it wouldn't be the beta cells, but what it is taking stem cells and trying to cajole them to make them turn into something that looks like a beta cell, functions like a beta cell, or actually perhaps functions like an entire islet of Langerhans, these clusters of cells, so that they can be then put into patients.
That sounds great, but I feel like you've answered our second question as well about what is cell replacement therapy and curing diabetes. But how is it linked to gene regulatory processes?
The purpose of looking at the gene regulatory processes is that, as I say, that well, stem cells by their very nature are cells that can become lots and lots of different tissues and different cell types, because that, particularly embryonic stem cells. So if you're trying to get one of these cells to turn into what is effectively an adult cell that has one particular function rather than lots and lots of different functions, um, what you want is you want it to actually stop being a stem cell and become the adult cell that you're after, whatever that may be. In this case, talking about beta cells or islets. So the last thing you want is for it to retain some of that stem cell like property, because stem cells are very good at dividing very, very quickly, and also turning into lots of different cell types. So what you find is actually that, and this has been done in various animal studies, if you put these cells into into animals to try and sort of effectively cure, in inverted commas, their diabetes, then what will happen is that often those cells will do that. They will actually be able to
Hmm.
prevent these animals from having, so it's usually in mice, so prevent them from having diabetes anymore. However, the downside is that the stem cells within that mixture of cells will often go on and form tumors and cancer in these individuals. So the last thing you would want as a treatment is obviously to be injected in cells that may well turn into tumors. So a lot of that is because these cells are unable to lose that stem cell property of being able to divide very, very rapidly. Um, and therefore, that's the last thing you want if you're trying to make a treatment. And nobody's managed to crack that yet. Um, so the prize is still there, I would say.
Now, thank you. I hope I'm pronouncing all these genes right. Quite complicated names. So the genes hexokinase 2, P85 alpha P13K, and SREBP1C. What is their significance, and do they have similar functions?
That's a good question. And you did pronounce them correctly, by the way.
Thank you.
They're not always the easiest things to pronounce. So hexokinase 2 is, I'll do them in the order that you that you mentioned them. So hexokinase 2 is an enzyme, so it's a protein that carries out a reaction in our cells, and its basic function is to phosphorylate glucose. And what I mean by that is glucose goes into our cells, and phosphate, particular chemical group, is effectively attached to that glucose molecule. And it's actually the first step in a 10-step process called glycolysis, which is actually the first step in beginning to break down glucose in our cells.
Respiration.
Yeah, exactly, and respiration to yield the energy from that glucose. So that's its function fundamentally, and it's in most of the tissues of the body actually as well.
Hmm.
So it's kind of all over the place. It's not in one particular organ.
Yeah.
And in diabetes, usually hexokinase 2 is not degraded quite as quickly as it would be normally. So it kind of hangs around in the cell a bit longer. And that's kind of made worse because in diabetic individuals, they're hypoglycemic. So they have high levels of glucose already. Um, there's really large amounts of glucose in the system. It's converted through the process of the reaction to glucose 6 phosphate. But actually, the process of glycolysis, this respiration process, as you mentioned, is actually kind of overloaded by the fact that hexokinase 2 is kind of in abundance, if you like, in the cell. So there's too many of all the intermediate molecules in that 10-step process of glycolysis.
Mm-hmm.
And what that does, it has a knock-on effect of having lots of different problems in the cell. So starts to trigger particular cellular stress responses, and also starts to muck up how the mitochondria that are in cells function. So you start to get mitochondria malfunction and not working properly. So instead of glucose being used to yield energy from it, what it's doing is kind of mucking up the processes in the cell, really, because there's larger amounts of this hexokinase 2 enzyme present.
Yeah.
So ultimately, that means that the cells begin to deregulate themselves effectively.
Would more hexokinase 2 mean less glucose is broken down?
No, so it's a process called glycolytic overload. So it overloads this glycolysis or respiration process. And it's made worse because, as I say, in diabetic individuals, they will have high levels of glucose anyway. So not only do you have a kind of overactive respiration process because you have more of this hexokinase 2 enzyme present, but equally there's more glucose present, so the whole system becomes overloaded. And the knock-on effect is that things are kind of knocked out of kilter, really. And as you probably know, kind of homeostasis is the key thing really for biology. And this is a good example of where it really gets thrown,
Mm-hmm.
and you end up with lots of cellular damage and cellular processes going awry. So that's the first one. So P85 alpha PI3K, or we'll call it PI3K for short, shall we? So that's phosphatidylinositol 3 kinase is what that stands for, but we'll definitely call it PI3 kinase. So that's another enzyme, and that phosphorylates other proteins in the cell, one of which is the insulin receptor.
Mm-hmm.
So, it's part of a process that the insulin receptor is involved in. It doesn't directly phosphorylate the insulin receptor. And if it's in beta cells where the insulin is acting, so the insulin's binding to the receptor and then triggering a cellular response,
Mm-hmm.
then that could be for releasing more insulin from the beta cell. If it's in other cells of the body, then what tends to happen when insulin binds to the insulin receptor is that you will have glucose transporters or glucose channels that are pushed out to the cell surface to allow those cells to take up glucose. So often insulin is described as kind of, it's the key to open the lock to allow the glucose into our cells. What's actually happening is that there's a whole raft of different cellular processes that present lots and lots of glucose channels to the cell surface, and once they're on the cell surface, the glucose can get in. So they're the processes that PI3 kinase is involved with.
Mm-hmm.
And if it's in a diabetic individual, often there's mutations in that gene, which causes mutations in the protein, and you get particular problems with this insulin signaling process. So it starts to kind of deregulate the insulin signaling process. So normally in the liver's kind of the hub of all the metabolic processes that happen in your body, really.
Mm-hmm.
So in the liver, the effects of insulin are usually to prevent glucose being produced. So it knocks out the process called gluconeogenesis. It reduces that process of making glucose from other molecules. And you increase production of fatty acids or fat molecules from non-fat molecules like glucose, for example. So that's a process called lipogenesis. So insulin normally does that. And when you have elevated levels of sugar, then both of those processes are activated. If this PI3 kinase enzyme is disrupted, though,
Mm-hmm.
the liver starts to make too much glucose. because it's not being knocked down anymore. It's not being reduced. Um, and as it's making too much glucose, that is effectively being pushed out into the bloodstream. So you end up with even higher levels of blood glucose, actually. And it's kind of seen as a bit of a precursor to type 2 diabetes, which is the form of diabetes where it's not that you don't make insulin, it's just that the insulin you do make becomes kind of ineffective. It's a process called insulin resistance. So kind of another way of putting it is the insulin's kind of got a limited action upon the cells that it should act upon because it can't carry out its normal function.
So does the P85 alpha I3K only have an effect if, example, a person has type 2 diabetes and then further he has the gene P85 P1 PI3K? Would it just be more serious in that case?
It can be linked, but as with most conditions, it's usually not just necessarily one thing that would be causing this. So there are mutations in PI3 kinase that are linked to type 2 diabetes. So yeah.
Okay.
And then the final one was SREBP1C, yeah,
Yeah.
which is steroid regulatory element binding transcription factor 1. So that's as the name suggests, it's a transcription factor, and they're as I mentioned earlier, they're kind of proteins that they bind to DNA and they regulate particular genes. So this isn't an enzyme like the other two.
Yeah.
What it does is it regulates a whole range of different genes that are linked to how cells utilize glucose and also fatty acid synthesis. So it links a little bit to some of the stuff I said previously that so glucose and fatty acid, in fact, most of energy metabolism, most metabolic processes can be deemed as malfunction in diabetes in certain instances. So this is a transcription factor that regulates this range of genes that are linked to glucose, as I say, usage and fatty acid synthesis.
Mm-hmm.
And there's actually been reasonably recent studies done on it's diabetic individuals that are in mice, so not in humans, but that the process of if they're obese or diabetic, so that there's metabolic stress that they're put under, and this is the same in humans as well. That can trigger this gene, so this SREBP1C gene to be activated. And normally, what would happen is that in individuals where this is functioning properly, you end up with an increase actually in insulin secretion, and you end up also with an increase in beta cell proliferation. So you end up with more beta cells in those individuals.
Mm-hmm.
And in the early stages of pre-diabetes, if you like, that tends to happen quite well. The problem comes when that stops happening, and you're not able to increase insulin secretion enough to actually have an effect. So under normal conditions, this gene is involved in transcription factors involved in regulating some of these processes. In individuals where that gene is not functioning quite so well, you don't get that process. So you don't really get as much of a kind of compensation, if you like, for the effects of the metabolic stress caused by obesity and diabetes. So that's how it kind of ties in with how diabetes can form. So if there are individuals, again, a bit like the last protein, if they've got mutations in that protein, it maybe doesn't quite carry out the function as well as it should do.
Great. I just wanted to ask, do you know how you said if the gene was malfunction or was not present, then there would be more beta cells proliferated? I thought beta cells produced a lot of insulin. Wouldn't that mean that it has to like work more better? Or does it not work because type 2 diabetes is not about like the amount of insulin, it's just about the cells not taking insulin?
So in obese individuals and diabetic individuals, or sometimes both, then what you have is effectively is metabolic stress because you have highly elevated levels of blood glucose usually,
Mm-hmm.
amongst other things. So what tends to happen in response to that is that the pancreas is kind of triggered and signaled to create more beta cell mass, if you like, so more beta cells, and therefore make more insulin in response to that elevated blood glucose level. So this isn't normal conditions per se, but it's normal in that the beta cells are compensating for this metabolic stress the body is under. Whereas when this gene doesn't work properly, that doesn't happen. So in other words, the blood sugar remains even more elevated than it would be otherwise as well.
Makes much more sense. Thank you. Moving on to the next question. What is the current research being done on diabetes?
So that's a very broad question, which I quite like actually. I think there's for me personally, I would say there's probably three main areas. So there's cell replacement therapies that I kind of touched upon earlier.
Yeah.
Particular ways of injecting or types of insulin, and immunomodulation. So that, from a cell replacement therapy point of view, there's so I mentioned stem cells previously, but actually other ways of looking at that is rather than using stem cells, to use something called IPS cells, or induced pluripotent stem cells. So they're actually cells, usually, well, in theory, they would be from the individual who has diabetes, and then therefore you're not taking them from somebody else. So therefore, if you transplant them back into that person, they don't need to have immunosuppression, which is what they would have to have
Mm-hmm.
if they had an islet transplant, let's say, which would come from other individuals. And to have the drugs that kind of suppress your immune system. Firstly, you don't have a very good immune system, which isn't great, but also the drugs have quite severe side effects. So if you could use IPS cells instead of embryonic stem cells, or indeed, other sources of islets like islet transplants, then
Mm-hmm.
it gets around all of that. And then linked to that as well is actually, even if we did use stem cells from other sources, let's say, and try to convert them into our beta cells or islets or Langerhans, is to try and encapsulate them in some way. Um, so there's a company called ViaCyte who are at the later stages actually of trialing this. So you have what in effect looks like almost like a wound dressing, so it's this small patch, and inside that is where all of the cells are,
Mm-hmm.
and you can put that under the skin of a patient. It can function as almost a kind of large cluster of cells, and then if something goes awry, you can take it out. So the nice thing about that is it's kind of a mixture of the cell biology bit, but equally kind of more techie side of things as well, in terms of how you would encapsulate these things.
Uh-huh.
Probably linked to the techie side of stuff as well is insulin injections. So even though as a technology, if you like, it's very old to kind of inject drugs into people, but using insulin pumps that are getting ever smarter and being able to detect constantly your blood glucose level and then release insulin as and when required.
Yeah.
And equally what those insulins are. So there are newer insulins now called glucose responsive insulins, or sometimes called smart insulins, that you can be injected with very high levels of the insulin itself, and it remains inactive until your blood sugar starts to rise, and then
Mm-hmm.
it becomes activated. And it activates in response to the level of blood sugar you have. So that combination of those things, I think, could be quite powerful. And then linked actually to the patches and the kind of encapsulation of cells, immunomodulation by using specific material that those patches are made from,
Mm-hmm.
that triggers your immune system, but in the kind of right way, if you like, to promote the correct response from the immune system. So there's people looking at materials based on
Mm-hmm.
extracellular matrix, which is the proteins that basically hold us all together. So they're the proteins that sit between all of your cells, and looking at making materials out of that rather than out of completely artificial material, in the hope that would be able to trigger a response that's a good response. It triggers kind of the immune system to kind of protect that patch rather than kind of attack it, if you like. So yeah, so they're the three things for me. I think the replacement therapy bit, types of insulin and how they're injected, and immunomodulation. But I think the really cool thing for me, I think, is that is it's this really interdisciplinary mix of subjects in a way, because it's not pure biology, but it's equally engineering problems as well. And that is definitely the way, not just this, but the whole of science, I think, is going.
Great. The patch device is super cool though.
It is, yeah.
If you had to invent something to manage diabetes, what would it be?
It'd be a patch device that's super cool. Somebody's already there already, but anyway, yeah, I think for me, probably That's a tricky one, but I think probably that what I just said actually, this kind of bioengineering and being able to do that in combination with manipulating cells as accurately as possible,
Mm-hmm.
because ultimately. The amazing thing about any form of multicellular life, actually, in fact, it doesn't matter, it can be single cell life as well for that matter, is the real kind of exquisite control that happens when cells develop.
Mm-hmm.
And we're just nowhere near that in terms of being able to do that in the lab. So a greater understanding of those processes, I think, to prevent some of the things that I mentioned previously about cells misbehaving, and linked with actually being able to deliver those cells into a patient without anything happening. So again, these patches, but it's quite crude in that you'd be sticking a patch under your skin, but could we do that in a better way? So that combination, I think, of engineering and cell manipulation, I think, holds the most promise, I would say.
That's actually super cool. So is insulin a hormone or an enzyme? If it's the former, how does it break down glycogen?
Insulin is definitely a hormone. So enzymes, as I mentioned previously, are biological catalysts. So they carry out reactions in our cells that otherwise wouldn't happen without them.
Uh-huh.
So yeah, insulin is a hormone, and a hormone by contrast is something that's made. It could be a chemical messenger type compound, or a chemical compound, or it could be a protein, like insulin is, but released by one cell that then goes and has some effect somewhere else in the body. So insulin, made by beta cells in the pancreas, but when it's released into the bloodstream, it has its effect all over the body. Adrenaline is another example, but that's not a protein. That's actually an actual chemical compound that has an effect all over the body, even though it's made in the adrenal glands. But insulin itself, it does have a link with glycogen, so it actually builds up glycogen. Not directly, the insulin itself doesn't do that, but it signals to the cells to activate glycogen formation. Glycogen is I'm guessing you know already because you've asked me the question, but glycogen is great big huge molecule that's made up of individual glucose molecules. So it's this great big branch structure that kind of is built up when, and I tend to describe this when I teach this in my classes, as kind of when times are good and the body's got lots of energy, then you don't need the glucose to break down to make energy because you've got plenty of it already. So you store it. So the glucose can be stored in glycogen, most of it in the liver and in your muscles, in skeletal muscle.
Uh-huh.
And then if you've not eaten for a while, then times aren't quite so good. So you start to break down the glycogen, and in the liver, the glycogen's is released into the bloodstream to keep your blood sugar up. The glycogen is actually not released at all; it's used up for muscle contraction.
Uh-huh.
So insulin affects the formation of glycogen by triggering a set of responses in the cell that build up this glycogen molecule. So that's what happens. So it does have a link to glycogen, but it doesn't directly impact glycogen. It sets off a set of processes that create the glycogen molecule from other enzymes in the cell.
So that's pretty much it. That was such an interesting episode. I enjoyed a lot, and that brings us to an end. Thank you so much, John, for being on this episode.
No problem at all. Thank you.
That's it, guys. That brings us to the end of today's episode, and I hope you enjoyed it a lot. And tune in for more episodes from the TechFest podcast. Bye.
Bye, everyone.